Focus of today is simple Mendelian genetics. So I'm first of all going to run through some of the vocabulary that you will be encountering and you'll need to understand the concepts in this section. So genotype and phenotype, those words sound very familiar. Make sure you understand the difference between them. We'll be using them quite a bit. So genotype is fairly easy to remember because it just means the type of genes that you have. And we're going to use capital letters to stand for dominant genes, and we're going to use lowercase letters to stand for recessive genes. So the genotype will be annotated with letters basically. Then phenotype refers to how those genes are expressed, usually how they appear. So that can be a color or a size or a shape. It's usually observable, not always, but usually. So in the drawing I've got a tall plant and a short plant. So the concept of being tall or short would be the phenotype there, and the letters beside it. So you're looking at the dominant letters, the tall being dominant, and the short, the lowercase letters signifying the short or the recessive form of that gene. Another important vocabulary word will be allele, which means an alternate form of the same gene. So if I were talking about the gene for height, then two alternates for that gene could be tall and short. So I would signify tall with a capital T and short with a lowercase t, just to show that I'm talking about the same trait, but I'm looking at alternate forms of that trait. Two other words you're going to want to make sure you understand are homozygous and heterozygous. So the prefix homo means same, and that just means the alleles in a genotype will be the same, whether those are dominant or recessive. So it could be two big T's in this case or two little t's. Heterozygous means different. Well, hetero means different. So the alleles are different. In this case, you have two alternate forms of the gene. So you have the allele for tall and carrying the recessive allele for short. The only thing I've added here is just alternate names. So homozygous could also be called purebred or pure breeding, and heterozygous could also be called hybrid. Okay, this next section we're looking at is called Mendelian Genetics. It's basically simple genetics. It's called Mendelian Genetics because of this guy, Gregor Mendel. And I'm not going to go really deep into the whole history of how he came to his conclusions. You can read about that. That would probably be a better use of time. What I'm going to talk about is his laws of inheritance. So Gregor Mendel focused on 7 different traits that he could distinguish in pea plants. I'm probably not going to talk about all 7. I used the T's earlier, so that would be for tall, so height of pea plants, and then we'll look at the color of the pea pod here. So basically he found that each trait had 2 alternate forms. So seed color could be green or yellow. And I did not have a yellow marker, so I'm just leaving that one blank. So basically, you'll find that he crossbred pea plants that were known to be true breeding for their particular trait. So here, this was a green pea plant. So all the pea plants in this lineage had always been green. They'd never been anything but green. you would consider it a purebred green plant. And then this one, yellow. So he took those two and he managed to cross-fertilize them, and see what happened. And he would call this his P generation, or his parental generation. And then he took the next generation of pea plants and he looked at them, and he called this his F1 generation, or his filial 1 generation. So his first family might be another way of thinking about that. And in this case, he didn't get any green pea plants. And that was quite surprising to him, because you would think that if you cross a yellow and a green, you're going to at least get some greens, or maybe something in the middle. So either one of those hypotheses could have been correct, but he didn't get any greens at all. So then he took a couple of these pea plants and he crossbred those ones, and he called the offspring his F2 generation. Now in this F2 generation, most of them were yellow, but several of them were green. So that green trait that was here in the P generation showed up again in the F2 generation. And what Mendel did is try to figure out, well, how did that happen? What is the mechanism by which traits get passed from parent to offspring? And that's what we're going to look at here. So let's first of all look at this chart here. This one has genotype, which I already identified for you as meaning the type of genes, okay? So in the type of genes that he had in the P generation, I'm going to go ahead and say he had purebred yellow. And notice I've made those Ys because we just know them to be dominant. Okay, so they're going to be capital Y. And then he crossed them. I'm not going to use G for green because it's a similar form of the same trait. So I'm going to use little y's instead. And I'm just going to make them little because they're obviously, he didn't have this word for them at the time, but they're obviously recessive to the yellow because they don't show up in the next generation. So that would be the genotype of the parents. In the F1 generation, we can say that for sure they've got a big Y because they're all yellow. But this parent only had little y's. So they got a big Y from this parent and a little y from this parent. Of course, Mendel didn't know that. All he knew is that their phenotype was yellow. And the phenotype here is green and yellow. Okay, so let's go back to the crosses that Mendel made and try to sort these out in terms of genotypes. So, this one being yellow, and Mendel chose that yellow plant because it came from a line of yellow plants. There'd never been anything but yellow pea seeds in that line, and so we would call it pure breed or pure breeding. So we will show that with two big Y's. And this one also was pure breed, green. Never been anything other than green in this lineage. So we'll call that little y. Now why am I calling that little y instead of G? It's a good question because it's green. You would think I would call it G, but no, it's the same. It's an alternate form of the same gene. So these are two alleles for the same trait. So for color. So in order to signify that I'm talking about the same trait, I just want to show that they're alternate forms of the same trait. So color. This one is yellow, this one is green. And that's how you do it. You just use the lowercase letters to show the recessive. Okay, so you might remember doing multiplication tables in elementary school. This is called a Punnett square, but it kind of works the same way. I'm going to take the alleles from this parent. So in other words, the two big Y's. I'm splitting them up because meiosis splits them apart, right? And so you've got two different possibilities for your gametes. Well, you don't. They're all going to be the same, but you could. And in this case, you've got little y's in the gametes here. Okay, so what we're going to do is just like a multiplication table. I'm just going to go, okay, big Y from this parent, little y from that parent. And then here, big Y, little y. Same thing here, big Y, little y. Big Y, little y. Okay, so we've got 100% big Y, little y. This would be considered heterozygous or hybrid. And it's not surprising that in the F1 generation then, Mendel saw that 100% of his pea plants were yellow from this cross. So, but we can look at this Punnett square and see that, yeah, of course they're yellow. We've got a dominant yellow, which will mask a recessive allele every time. So you're never going to see the green, but it can still be passed down to offspring. So let's do another Punnett square here. And let's look at what happened in the F2 generation. So Mendel took two of these pea plants and they had this genotype, so big Y, little y, and two of them, and he crossbred them. And that's what he was doing here. So this individual is going to have big Y, and of course it's going to be yellow. I'll just keep track. This individual is going to get this big Y, this little y. Oh, it's also going to be yellow because it's got the dominant allele. This individual is going to be big Y, little y. Also going to be yellow because it has that allele, that dominant allele. But look what happens here. We get a little y from that parent and a little y from that parent, and we've now got green. So we've got a 3-to-1 ratio, green to yellow, and that's what he saw in the F2 generation, roughly a 3-to-1 ratio. Okay, I'd like to try to apply this to another trait. This is a huge simplification of human eye color, which is not controlled by only one gene, but we can simplify it and look at it in the same way that we look at pea plants. So we know that brown eyes are dominant over blue eyes, and by blue that can be any kind of shade like green or blue or hazel. It just means anything non-brown. Okay, so a person could be homozygous dominant, meaning they could have the same dominant alleles and have brown eyes. You could be heterozygous and have one allele from each. Like you could have got a brown-eyed allele from your mom and a blue-eyed allele from your dad. Your phenotype is still going to be brown eyes. However, you could pass along the blue-eyed allele to offspring. Or you could have two recessive alleles, or another way to say that is homozygous recessive, and you could have a phenotype of having blue eyes, and you would have a recessive genotype. And we can apply the Mendelian genetics to something like eye color. So again, we can just make ourselves a Punnett square. And we can think about meiosis. I can do any cross here. I'll probably just do a few. So what if a person who had brown eyes, that was their phenotype, but they were carrying a recessive allele for blue, had a child with a person who had blue eyes. So here we have a brown-eyed parent, well, let's call him Dad, and a blue-eyed parent, let's call her Mom, okay? And we want to see what color eye colors her offspring could possibly have. So we do the same thing as we do for pea plants. This is simple Mendelian genetics. So a big B from Dad, a little b from Mom. In this case, the same thing. A big B from Dad, a little b from Mom. Here we've got a little b from Dad and a little b from Mom, and the same thing here. So we would say that their offspring have a 50-50 chance of having brown eyes or blue eyes. 50-50 in this case. Let's just try another one. So this time Dad, well let's leave Dad. He's still heterozygous. And let's say Mom is also heterozygous. I'm just going to erase these. You could follow along if you have some scrap paper. Okay, so first of all, let's say, okay, Dad is going to give a big B here for brown eyes, and so is Mom. So this offspring is going to be heterozygous dominant for brown eyes. Now here, Mom is giving a big B, but Dad's giving a little b. This person still has brown eyes, but we would call them heterozygous. Now this person, getting a B from Dad and a little b from Mom, heterozygous. But look what happens to this offspring. They're getting a little b from Dad and a little b from Mom. So their phenotype is going to be blue-eyed. So it explains why two parents with brown eyes can end up having a blue-eyed child. We might be asked a variety of different questions on a problem like this. We might be asked, what's the probability that the child would have blue eyes? Well, you could express that in a number of ways. You could say there's a 1 out of 4. You could say there's a 25% chance. If you were asked for the ratio, it would depend on whether you were being asked for the ratio of phenotype or genotype. So if you were being asked for a phenotypic ratio, you would have 3 brown to 1 blue. If you were being asked for a genotypic ratio, though, your answer would be quite a bit different because the genotypes are different. It's not a 3-to-1 ratio for genotypes. You've got 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. So the only way to get comfortable with Punnett squares is to practice them. And this simple Mendelian genetics really is foundational for going on and doing more complex genetics, which we'll be doing over the next few weeks. So lots of practice. And I'm going to leave it there for now. And tomorrow we're going to talk about what happens when you cross 2 different traits at the same time.